This is the first video for the types of cells lecture, and for this we're going to be reviewing prokaryotic and eukaryotic cells. The big difference right off the bat is that prokaryotic cells are smaller and lack internal structure or compartmentalization, whereas the eukaryotic cells are larger, they have a nucleus and other internal structure. And those internal structures are called organelles, which literally translates to tiny organs. But before we dive into the details of prokaryotes and eukaryotes, I want to first talk about bacterial morphologies and arrangements. So this is unique to bacteria, and bacteria are prokaryotes. Therefore, they are also unicellular, meaning the entire bacteria is one single cell. And in lab, we're going to be looking at different cells under the microscope. And whenever you're looking at cells under the microscope, you always want to look at their morphology and arrangement. So morphology is a fancy word for shape. And so today I'm going to use it interchangeably, the terms morphology and shape, but after this video, I will constantly just say morphology. So there are three major categories of morphologies, and that's going to be circular, rod-shaped, and spiral-shaped cells, okay? But of course, we need science terms for that. So circular or spherical-shaped cells are called coccus. Okay, cocci for plural. So if you're looking at a single round cell, you would say it is coccus. If you're looking at hundreds of round cells, you would say they are collectively cocci. Okay, if they are more rod-shaped, okay, then the singular term is bacillus. The plural term is bacilli. And you can see that in the second row of cell shapes. The third major category is spiral-shaped cells, but of course, got to make things a little complicated for you. There are actually two separate terms depending on the specific shape of the spiral. So spirillum, which is the second from the bottom in the table that you see, spirillum tend to be thicker and have less spirals, less turns, okay? But if you compare that to the spirochete, they tend to be long, thin, and have more coils. There's the term I'm going for. So if you compare the illustration of a spirillum to a spirochete, you can see that there are more coils in the spirochete than in the spirillum. And I think the actual image of a spirochete versus a spirillum really kind of drives it home, okay? So the spirochete, again, tends to be a little bit longer, but much thinner and several more coils than the spirillum. All right, two other shapes that you will encounter during this class are vibrio, which is more of like a comma-shaped cell, so comma or curved. And then coccobacillus, I think, causes myself and others to overanalyze the shapes of cells, but there are some that do look like squished bacillus. So they're not quite round, but they're not quite rod-shaped. So we just merged the two names together and we call it coccobacillus. But for the purpose of my class and our labs, don't worry about using coccobacillus. Just say they're either cocci, they're bacilli, or they're one of the two spiral-shaped cells, okay? And when you're looking at something under a microscope, or if you're looking at a prepared image, if you're virtual, if you look at the cells and you go, oh, those are circular, then go with coccus. If you look at the cells and you're like, that's not really a perfect circle, then go with bacillus, okay? I will always try to provide slides that aren't overly tricky because microbiology is hard enough without questioning your shapes. So I will try and make sure it's very obvious if it's circular or not. But please don't worry about, is it coccobacillus or just bacillus? Okay, I'm not going to be mean and make coccobacillus even an option on your quizzes or lab exams. Okay, so these are your morphologies or shapes. The next thing you want to look at is how they are arranged. And basically, whenever you're looking at a prepared slide or a slide you've made under a microscope, you want to look for where there's some spacing between the cells, because especially when you're learning how to do staining, people tend to, and this myself included, tend to put too many cells on the slide. And so everything looks like they're grouped, like that all the cells are in groups. And sometimes that's just because you got a little carried away when you're putting cells on your microscope slide. So you always want to look at kind of some nice spacing, when there's nice spacing in between the cells, like they're not all smushed together. And if you notice that the cells tend to kind of hang out in isolation, then you would say it's a single coccus or single bacillus, again, depending on the morphology of the cell. Okay, but you'll just say it's a single cell. Now, if you're looking at your slide and you constantly see the cells paired up, then you use the prefix diplo, for di for two, diplo. So if there's two circles, you would say diplococci. And I apologize, on this slide here, I think it's a little misleading. Diplococci are just those two purple circles. For the two bluish bacillus cells, you would say it's diplobacilli. Okay, so diplo should have been the only thing I had typed there. So diplococci or diplobacilli. For tetrads, you will only see coccus cells in tetrads, okay, because of the planes of symmetry. Bacterial cells that are coccus can divide by binary fission along multiple axes, okay. Let me try and sketch this really quick, and I'm using my mouse rather than a pen, so it's gonna look like a small child did this. So for a coccus cell, pretend that's a perfect circle, right? They have planes of symmetry, right? Pretend that's in the middle, sorry, y'all. And so the cells can divide horizontally or vertically, okay? But with bacillus cells, okay, give it use your imagination. Bacillus cells can only divide along the end, okay? So they can only grow end to end. Okay, they cannot grow like stacked on top of each other. Not happening with bacillus. Okay, so that's why I didn't put any bacillus cells here at the tetrad, because again, you can't get them growing on top of each other. Okay, that's a no-go. So if you see tetrads, it had better only be of cells that are coccus. Now, if you see that all the cells tend to hang out in clusters or groups, then you use the prefix staphylo. Okay, now again, because of this planes of division issue, you will never see staphylobacillus as the arrangement. Okay, because again, bacillus cells cannot divide along that long end. So if you notice that the cells are arranged in lines or chains, then you use the prefix strepto. So this chain of cocci cells, those are called streptococci. The bacillus cells, you would describe this arrangement as staphylobacilli. Okay, so again, you always put the arrangement prefix first, and then you write down the morphology. Okay, now you can ignore this sarcina. We won't ever encounter this. I meant to delete it and I completely forgot, so I apologize for that. You will not need to know about that. Okay, so again, remember, arrangement goes first and then the morphology. So you might have diplococci or staphylococci or streptococci, but you can only have one arrangement. Okay, and that's why as you learn to stain, you have to learn how to not take too many cells, or else you can confuse yourself when trying to determine the arrangement. All right, y'all, that's the end of this video. Please let me know if you have any questions.